Welcome you all to this Cryo-EM course. And the last class I discussed about the three aberrations. One is chromatic aberration, then next one is astigmatism, and then third aberration, the spherical aberration. Now there is a one very important aberration we are going to discuss, that is called the coma. What is that coma? It is a bit difficult to explain this coma shape in this kind of 2D system. During that practical class I will show you how to correct that coma, but today I am just going to explain using some diagram how it should look like. Let us take a look how this coma aberration look like. So now we are talking about that coma aberration, right? If we just consider that there is coma aberration, again it is a off axial aberration. This is a off axial aberration. So how this off axial aberration is happening? There is a two aberration I talked about, that chromatic aberration and the spherical aberration, both are on axis. Now astigmatism is off axis. Now coma is the another one, that is called the off axis aberration. How this off axis aberration is happening? You consider that okay, there is a some kind of deviation of the, so there is a two possibilities, that okay, when you have a filament, so filament tip we are always expecting that electric current, it is going like that. So there is a one possibility that okay, there is a filament is tilted, so it is tilted, right? So there is another possibility is that when we are inserting the sample is tilted somehow, and from the sample it is coming. There is couple of possibilities are there, that why there is a tilt and this problem it will come. Now you consider that something is a very perfect situation and all the rays are coming from the center. So if all the rays are coming from the center, how this will come all the way, and it will come all the way here, and it will give us an image. So it is not like that one single rays it will come, the optical axis, this is the optical axis. Now rays will come and merge here, right? So this is the perfect scenario where rays are coming like this and giving us an image, and our point source, or if we just consider our sample and our image, everything on the axis. Now if there is a deviation of this one, if the point is shifted like that, and the magnetic field whatever it is, so if we have a magnetic field, if we just consider this is our magnetic field, so this is our magnetic field and there is a no change in the magnetic field, what will happen? So first ray, so first we have to draw this is the image plane, right? So first ray it will go and converge in one particular point, this is the optical axis you consider. So when it is shifted, now I am considering this is my optical axis. Now based on this optical axis, rest of the ray diagram we have to draw. So now this is the first ray, and here this is the first blue ray it is coming. Now this ray it will go and it will converge somewhere here, and this ray will also come and merge in this particular point. Then what about the rest of the rays? So if I just erase this black rays, it will be easier to understand, that's why I am just erasing that inside rays. Now if I just take the blue ray again, and how it is going to converge, so now next ray it is coming somewhere here, and this is the other ray. and both of them are going and converging somewhere here. Now you can see this should be the optical axis, and if we just consider there is a optical axis something like that, and the rays are coming and after converging all are merging in one particular point here. So if this is our optical axis, circle center will be here. This is the optical axis, and this will be the brightest spot. If we just decrease the beam diameter, so there is a one possibility that okay, there is a very small beam diameter, and then beam diameter can increase like that. So if we decrease the beam diameter, it will come to the center of that screen, or if we just increase, the beam diameter will start from this center and enlarge it. In this particular case you can see it that okay, so this is the optical axis. We are expecting the brightest beam in somewhere here, but rest of the ray, one ray is merging here. Now if there is a more rays are coming, if there are more rays are coming out, so they will come so those rays, it will come and merge in this particular point, and same thing it is going to happen. So now you can see there is a crossover point, it is coming here from the some rays, and there will be a brightest spot will be here, and then this spot will start spreading. So if we just want that okay, there will be a center, and then from this center the beam will start increasing and beam will move like that. In this particular case that is not going to happen. It will start from some one particular corner, and then beam will start spreading like this. Beam will start spreading like this. So you can see that it is not, that beam will not started spreading from the center and it will enlarge like this. So it is starting something like this. So it has a coma shape. So how this coma look like? So there is a coma shape, and that is basically a aberration which is going to affect our images. It is going to lose the signal, and our beam sometime look a different shape or elliptical. So when we are talking about the signal, we are talking about that okay, if we see the power spectrum, there is a center very bright spot, and then there will be a minima and maxima. So if we just consider this blue is the brightest spot, so there will be a maxima and minima, and it will be a very circular ring. So if we have a coma aberration, what will happen? You cannot see those rings anymore in your power spectrum. You will see only a center bright spot. That means there is some kind of alignment issue, and our signal is so poor, it is affecting because of the coma. Let's take a look how this coma aberration look like. So this will be better to show you in a image. Here if we just go to our power, so this is our astigmatism. Now this is the of axial coma aberration, and when there is a coma aberration, your coma shape, it will be spreaded like that electron beam, and this electron beam. So if you look at this spread, the spreading is starting from one particular edge. This edge may be this or that side, but it is not coming from the center. Whenever you will try to spread the beam, beam will start from one particular side. Now we are talking about the distortion also. There is a three types of distortion we can see it. One is pincushion distortion, and one is the barrel shape distortion. So in some lens system, magnification increases as you go further off axial. So if you are off axis and you are constantly going and changing the magnification, you can see your beam is spreading in a different way. So that is two types of distortion. You can see it, one is pincushion distortion and one is a barrel distortion, and you can see a spiral distortion also. If you change the magnification, your image will rotate in a different way, and you will get this kind of shape when you will rotate that. So sometime that in a pincushion distortion, you can see that okay, corner of the image is has a different magnification, center has a different magnification. So same kind of problem you will get it in the barrel shape or spiral distortion. So if we want a perfect imaging system, most of the time it is better to just align those microscope properly. So electron beam should come through the optical axis. So here I am showing you a 5 different types of distortion altogether. So where you have a spherical aberration, in spherical aberration you can see that whenever rays are near the lens, they converged in a different position. Where rays are away from the periphery of the lens, they are converging in a different point, and that is called the spherical aberration. Coma is a off axis aberration, where the rays are the optical axis is completely changed, and then some of the rays is converged here, some of the rays is converged in a different plane, and you always have a coma shaped beam. And then there is a astigmatism. Astigmatism is something like that where your electromagnetic field is different in your lens system, and it is going to affect your image, and you can see a elliptical beam, you cannot see a circular beam anymore. Now curvature of the field, that is something connected with your lens system and the other alignment also. All together you will get a different type of curvature, where your edge has a different magnification and center has a different magnification. That we already discussed about that, pincushion distortion or barrel distortion or S-shape distortion. So those are the distortion is involved with the electron microscope. Most of the time we have to correct those distortion when you will do the actual imaging. Now go back and discuss little bit more about that, how image formation is happening in our system. If we don't have the any kind of distortion, how we will get that image. As well as one important part is this, that we have to consider that okay, for the biological science sample, how can we do the imaging. In the later stage, whenever we will discuss about the sample preparation, we will talk about these things in details with our perfect images. But before that, let us discuss little bit about how we will image the biological sample. Then it will be easier for us to understand why do we need to regulate the electron beam during our imaging. So if we just consider any biological sample is made by carbon, hydrogen, nitrogen, oxygen and sulfur, and there will be some metal ion here and there, but mostly if you just consider that carbon, hydrogen, nitrogen, oxygen and sulfur, those are molecular, it is a not a very heavy atom. So their molecular weight wise, it is a very light. So if we are considering the scattering, if we remember that when we are talking about the scattering, so nucleus is there, and then there will be a orbital. So then orbital, in different orbital, electrons will be there, and sometime electron will come and go back, that is called the backscattered electron, right, backscattered electron. Or sometime electron will directly collide here, and then this electron will go out, that will be the secondary electron. Most important case, that okay, in transmission electron microscope we are interested about that when there is an electron it is going through this one, there is no deviation, nothing, because it is far away. So if we just consider that, okay, no, this is not the right one. So if we just draw an electron somewhere here, because there is no electron here, it is an unscattered electron beam, and most of the electron, it will go like that through the atom. And because these are very light atom, it is not a heavy atom, and biological science sample, most of the atoms are very light atom. And now there is some electrons are going closer to this electron, and then there will be a deviation. So there will be a deviation. So this is unscattered electron beam. We already discussed about this, and this is scattered electron beam. So now whenever we are getting an image, we are considering that unscattered electron beam and the scattered electron beam. Now when you have a heavy atom, we can get more scattered electron, and that is good for our signal. We will get a very good image and contrast will be much better. But when our atoms are very light atom, and there is a scattering, this scattered electron beam is very nominal, so that will give a very low contrast. So that is an important part, that whenever we are talking about the transmission electron microscope, so there is a two things are coming. One is for material science and one will be for bioscience. So similar kind of term, that okay, either 120 kV or 200 kV or 300 kV term we are using for material science and biological science sample. So material science sample, there is a, most of the time we are using a very heavy atom, and also those are not very soft material, those are not damaged by the electron beam. So we can use a huge amount electron dose, and that huge amount of electron dose, it will give us a very good signal, very good contrast. But for in our biological science sample, that is one biggest problem, because it is made by carbon, hydrogen, nitrogen, oxygen, sulfur, and those are very light atom, and the scattering is very low. So here if we just use a very strong electron dose like material science, this kind of sample, then it is going to damage our biological sample completely. In second class I already discussed about that electrons has a damaging property. It is going to damage the biological science sample. So we have to be very careful when we are talking about biological science sample. So how do we control the electron dose, and what is the good electron dose for our imaging, that we should know before we use our sample or insert our sample inside the microscope. So we will get a different types of microscope. In the material science or chemical sciences, we will get a many microscope. Can we use those microscope for biological science sample purposes? Yes, we can use it, but there will be a certain protocol that we have to follow, and then we have to use a reduced dose. And how we are going to reduce the dose, and what is the good dose for the biological science sample? That is the next question. Before talking about what is the good dose, let us see how we can image our biological science sample. There is a two methods that we are going to discuss later stage when we will discuss about the sample preparation. But for now, you just remember there is a one, it is called the negative stain. One, it is called the negative staining. Negative staining room temperature sample. So this microscope we can consider that whenever we are using the negative staining, so most of the time we are using uranyl acetate or uranyl format heavy metal staining sample. So those heavy metal staining sample most of the time we are using at the room temperature, but samples are the biological science sample. When we are using negative staining sample, almost 70 to 80% biological sample is dehydrated. Okay, 70 to 80% samples is completely dehydrated. So here we can use little bit strong electron dose. But what about the cryo electron microscope? If we talk about cryo electron microscope generally we have to use a very small amount electron dose, because in this cryo electron microscope we are not using any negative staining. Okay, sample will stay in the buffer. You can consider that sample will stay in the water, and then your biological science sample. So that sample may be your protein or your DNA or your RNA, or there is a DNA protein complex, RNA protein complex, like that, and all that has carbon, hydrogen, nitrogen, oxygen and sulfur. So those are the component, and it will stay in the water. So water, it will stay as a liquid form? No, it will form a amorphous ice. Within that amorphous ice our biological science sample will be embedded in that amorphous ice. Now when there is a ice or amorphous ice, so within that sample if there is a very strong electron dose, that ice will start melting. If ice will start melting, sample will disperse, sample will be not fixed. In cryo condition we are using liquid nitrogen and liquid ethane, and we quickly plunge freeze the sample in liquid ethane. When you will plunge freeze that sample, it will just form a thin layer of ice, amorphous ice, and protein will be embedded in this amorphous ice. When protein will embedded in the amorphous ice, we are using a electron dose to image that sample. Now we cannot use a very strong electron dose. If you use a strong electron dose, ice will start melting, as well as the biological science sample, it is not in a dry condition. If it is in a dry condition, that strong electron dose cannot damage that much, because sample is already dried, it is a dehydrated. But when it is not in a dehydrated, in cryo electron microscope, the sample will stay in a physiological condition, near physiological condition, because sample will be completely hydrated. Now if there is a very strong electron beam is going through the sample, it will remove the water molecule, it will change the conformation of that DNA, protein or RNA, and that is going to destroy the three-dimensional structure of that sample. That is the reason, first thing first, we have to control the electron beam. So here if you look at in any type of negative staining or cryo, we are using, it is called grid for sample preparation. What is that grid? Grid is a circular thing. In that circular thing, this is a metal bar. It made by copper or nickel. Most of the days nowadays we are mostly using copper grids. When there is a copper, this is a metal bar, you can tell that this is the copper, made by copper, and it is a overall diameter, it will be 3.05 millimeter. I'll show you some picture of some images of that grid, how this grid look like. Now in this grid there will be this kind of metal bar. This individual square, those squares are empty. So you can see one small square, if I just target one small square, it is empty. It is completely a square and it is empty, nothing is there, and this is the copper bar. This is the copper, made by copper. Now on top of that, what is added? On top of that there will be a thin layer of carbon, it will be coat in entire grid. So I am just showing you here one square, but it will be all the squares. will be coated with a carbon layer. So there will be a very fine plastic coat, and on top of that there will be this kind of carbon layer at top. So now if you look at, the carbon layer will be there. And now if you just pay attention, so here I am just showing one square. Now for the cryo sample, if you look at this particular square, what you can see inside that square, so it will be better to draw this one because this is the boundary of that metal, and carbon is blue, this is our copper. Now if you look at that, here we have the carbon, here we have the carbon, all over carbon it will be there, and this is the amorphous carbon, it will spread all over. Now inside that one, what we can see, there is a uniform sizes hole, it will be there, and that will be the empty region. So it will be something like that. So if we just remove your carbon, it will stay. So this is the copper, this is our some hole, it will be there, and carbon, it will be outside of those holes. Okay, so there will be some carbon outside the hole, and this is the area where we are going to do the imaging. This hole we are targeting for the imaging purposes. So individual hole, so hole means, that is, if you just look at in AFM, that okay, atomic force microscope, if you just look at this, will be a hole something like that. Now whenever you will add your biological science sample, so it has water and sample, it will go inside that hole and it will stay inside there as a liquid form. Why it will go inside and stay in the liquid form? Because this is the hole, due to the surface tension it will hold a small amount of sample. Then when you will plunge this, this will be frozen, this will be frozen and your sample will be fixed. Now when we are going to do the imaging, what we are targeting? We are targeting this individual hole. So if you look at this individual hole, what we have there, if you look from that, there is a amorphous ice. As I mentioned that if we just image in this particular area, there is a amorphous ice, and within that amorphous ice sample is embedded. Okay, and the beam will pass through this one and give us an image. So now if you look at this, your beam diameter, when you want to image one particular area, you have to be very careful about the beam diameter. If beam diameter is very big like that, what is going to happen? It is going to expose all the holes. So here you are just targeting one particular region of that particular hole, and you are trying to get the image and get the sample. So now if your beam diameter is very big and it is going to expose all the holes, it is going to damage the individual hole and ice will melt from all these holes. So now if you just target one particular hole, so you have to maintain the beam diameter in such a way it should not expose the entire grid. That is the reason we have to control our electron dose when it will go through the sample. But same time we have to maintain our beam diameter. If beam diameter is too big, it is also going to damage the entire grid and we will be unable to get any good data from that system. So what is next then? Let us see how we can control our beam diameter and electron dose also. So let us go and do some ray diagram and see how this image formation is happening. Last class I already discussed about that focused, under focused and defocused. In all the cases you can see I am just using different kind of magnetic lens current and magnetic field to control the electron beam, and I am converging the electron beam as the different point. If you just remember that last class I discussed that, okay, so in one particular case it is converged here, this is called the focused beam, and one particular case electron beam is converged somewhere here, the below the plane, and that is called the under focused image. This is called under focused. Okay. and other one is over focused when we are using a strong magnetic field and the crossover point is going up and it is going like that. This is called over focused image. Okay. Okay, now you can see why I am repeating this thing. So you can see using that different magnetic field we can get a different kind of crossover point. This crossover point will be up or down based on our magnetic field. So let us see that, okay, if we are using a different kind of magnetic field, how it is going to affect. So in this particular case, whenever I am talking about the focusing the electron beam, especially for it is after interacting with the sample, how it is focusing and how we are getting the first image in the back focal plane. So back focal plane is the plane where we are getting the diffraction pattern. So in back focal plane the electron beams are merged, so it may be focused, under focused or over focused. The three things are there. Now, so whenever we discuss those things that focus the beam, it is electron beams are coming from the objective lens and it is already interacted with the sample and then we are getting those electron beams and that is coming as a image form. It may be in a back focal plane or it may be in the image plane. Now, what I discussed in the last slide, I discussed about that we have to control the electron beam diameter and the electron dose. Electron beam diameters means we can spread the electron beam and expose the entire biological sample and we can damage the entire sample, or we can just shrink the electron beam. So you can see that if we just consider that from the electron source 100 electrons are coming. Now 100 electrons, I spreaded that electron beam. If I just spreaded the electron beam, we are getting this big area where that 100 electrons is distributed. So we have a very big area. If you just consider that 100 angstrom square, it is the total area and the electrons is distributed. So in that particular case, total dose will be very less because 100 electrons is distributed within 100 angstrom square, but you know it is going to expose most of the sample. When I was drawing those hole, that diameter of this hole, it will be only 2 micrometer. So there are different types of grids are available where the distance between these holes will be very similar. So you know the diameter of the hole and this distance. So sometime this distance it will be 1 micrometer, sometime this distance between two holes will be 2 micrometer. So both the distance are very important. Why both the distance, both of these parameters are very important. Why it is so important, that we will discuss in the later stage. But just remember that, so this whole diameter, there are 3 different diameter you can get it, that 0.6 micrometer, 0.6 micrometer or 1.2 micrometer and 2 micrometer. So from here you can see that whenever we need a beam diameter should be within this range. It should not be too big. Our beam diameter cannot be 10 micrometer, 20 micrometer or 30 micrometer. If it is a 20 micrometer, 30 micrometer, that is going to expose the entire grid and entire biological sample. So what I was telling that, okay, initially if we have a very big beam diameter and if we have 100 electron, and if you just consider that total beam diameter is this big, and so total area of this one will be 100 angstrom square. Okay, and then total electron is 100 electron, then definitely dose will be less. Now we want to decrease the beam diameter because we need a beam diameter this size. So when I am making a beam diameter this size, and if we have 100 electron, all this 100 electron is concentrated in this diameter. Now you just consider this is a something 4 angstrom square or 10 angstrom square. Within that 10 angstrom square you are keeping 100 electrons. that per angstrom square it is a 10 electron. So here you can see it is depending on the beam diameter also. If you decrease the beam diameter too much, your electron dose will be high. So somehow we have to control this amount of electron, how much electron is going and going through our sample, how we are going to control that. So that control we can do it with using our C1 lens and C2 lens. I am just repeating the same thing. C1 lens is connected with your spot size and C2 is your intensity. Okay. So how can we control both the electron dose? So you have to go with a ray diagram. So here you can see both the electrons are coming and merge in one particular point, and this is my C1 lens current. Then we are using some kind of C1 lens current, and if I just use a comparatively less C1 lens current, what will happen? Electron beam will go and converge in one particular point. So here you can see this is called the weak lens, that is we already discussed weak C1 lens. We can say this is a comparatively intermediate condition. Now if we are just using a strong magnetic field or more strong magnetic field, what will happening here? You can see there is a crossover point. This crossover point is going up. When there is a weak lens system, your crossover point will be here, which is very close to the next lens or image plane. But if you are gradually go up for the strong magnetic field, you can see the crossover point is going up, and these rays, you can see that okay, these rays will go, this ray will come like that, right, for the next lens our C2 lens here, right. So here you can see that if we are using a very strong magnetic field, your crossover point it is going up. So in this particular case, when you have a strong lens current, that is your spot size is changing. This is your spot size, and spot size is changing, and this crossover point, that if we just talk about that crossover point, is going up here. This crossover point it is going up. So generally the spot size we can use 1 to 11. So if we are using spot size 11, that is means a very strong magnetic field, okay, and the crossover point it will be very up close to the lens, C1 lens, and your beam will be spreaded, it will be spreaded too much. So if you want to spread the beam too much using the C1 lens, you should use a very strong magnetic field, that means very high spot size. So I'll stop it here. In next class we'll discuss about how the C1 and C2 lens working together to control the electron dose. Thank you all.